In this video, we will explore the genetic differences between us and how they arise. Person A has 2 copies of the gene for eye color. The copy from her mother might code for blue eyes. The copy from her father might have a slight difference in sequence and code for brown eyes. Since both sequences code for the same protein that affects eye color, they are both genes for eye color. The different versions of the gene coding for different versions of the protein are called alleles. Person A has an allele for brown eyes and an allele for blue eyes. Another person B may have 2 alleles for blue eyes. This is a simplified example where we assume only one gene affects eye color. In reality, a trait is often affected by multiple genes. Suppose person A has blue eyes. The pair of alleles they inherited for the gene is their genotype for the gene. So an allele for blue eyes and an allele for brown eyes is their genotype. The actual trait we observe, here blue eyes, is the phenotype. The phenotype is affected by both the genotype and the environment. A location of the genome where we find differences in sequence is called a variant. These gray rectangles represent versions of the gene for eye color. Some of them are alleles for brown eyes, while others are alleles for blue eyes. Within the eye color gene, we may find that base pairs 100 to 120 vary in the sequences we compare. This region is a variant site. A variant is a location of the DNA sequence where we find differences between individuals. Recall that alleles are different versions of a gene in which there is variation. The term allele can refer to the entire gene sequence. The entire DNA sequence coding for blue eyes can be called an allele of the eye color gene. But when you use bioinformatics tools, you will find that some tools use the term allele to refer to not the entire gene, but instead just a few nucleotide bases which actually vary. That is, the sequence at the variant region. Let us recap the differences between gene, allele, and variant. A DNA sequence producing a particular protein affecting eye color is called a gene for eye color. A particular version of this sequence producing a particular version of the protein giving, say, blue eyes is an allele. Another allele of the eye color gene would have a different sequence, giving, say, brown eyes. The exact location in the DNA sequence where nucleotide bases vary is called a variant. We have 6 billion nucleotide base pairs in our genome. How many differences would you expect between you and another human? We share 99.9% of our DNA with other humans. This means that there are 6 million different nucleotides on average between any 2 of us. We share 99% of our genes with chimpanzees. The 1% difference makes us human. We share 50% of our genes with banana plants. So what causes new genetic variation? When DNA is copied, there is a small chance of error. We saw that the new strand is synthesized by complementary base pairing. Sometimes an incorrect base pair may be formed. Instead of an A pairing with T on the template, the new strand may have a G. There is now a difference between the new complementary strand with a G and the corresponding old strand with an A. In addition to nucleotide replacement, replication errors can add or remove nucleotides. The new DNA strand may slip while replication occurs. If the new strand slips, an extra nucleotide may be included. This is an insertion. If the template strand slips instead, there may be a missing nucleotide. This is a deletion. Such errors occur about once per billion nucleotide base pairs. This may seem accurate, but we have 6 billion nucleotide base pairs. Every time a cell divides and DNA is copied, an average of 3 errors are introduced. DNA can also be damaged by exposure to certain chemicals or radiation. Such damage also changes the sequence. If such changes in sequence occur in reproductive cells, we may pass these new variations to our children. All of us have many inherited variants as well as some new variants that are unique to us.